Good morning to some of you guys as well, since this is being broadcast across multiple time zones. My name is Sesha Tekor. I'm the technical specialist for high content screening solutions, and I welcome you all to the Connect to Science high content screening user group meeting this week. With me are my colleagues, Brent Sampson, Victoria Thane, and Matt Coughlin. And we, as a team, support the North American high content product portfolio, and we'd be happy to answer any questions that you might have now or down the road as well. Historically, the user group meetings have always been about our user and prospect base. And more importantly, these events are largely focused on highlighting some of the excellent work, that one of which you'll hear today, done by our scientists in the high content, using our high content screening platforms. And I personally hope that this week you would be able to see the value and the true power of high content technology for your own needs and workflow. It's actually been a great forum for all of us to bring the community together, the Helomics community, so to speak, so that everybody can share the value of the Thermo Fisher / Helomics high content solutions across the board. As a lead off today, I'm proud to introduce a friend. I consider him a mentor and a former colleague, Dr. Collins, Dr. Mark Collins. I just want to start off by giving a brief bio of Dr. Collins. Dr. Collins received his undergrad degree in applied science from the University of Wolverhampton, UK, and he had his PhD in microbiology from the University of Surrey, UK. Following his postdoctoral work, he's always pursued his passion, which is both looking into artificial intelligence and machine learning. And he used that to be a part of several drug discovery companies, informatics and bioinformatics functionalities across global pharma. And he was also instrumental in starting 3 startup companies during that process. Back in 2001, Mark migrated to the United States, where he joined the company called Helomics. And Helomics, as you probably already know, invented, coined, and patented the term high content analysis back in 1996. In 2001, when Mark joined Helomics and became a part of that team, he was instrumental and played a very pivotal role in establishing high content cell analysis market. And he built it and he commercialized it for the team. And that included several key informatics and bioinformatics products as well. This is where I came across Mark when I interviewed for a position back in 2005 as an application scientist. And Mark was one of the few who interviewed me. And honestly, my presence today in this company, I still owe a lot to Mark for taking an interest in me and making me a part of the Helomics team. So after leaving Thermo Fisher, Mark clearly focused on developing and commercializing informatics solutions for clinical and translational research. And he did that through key roles in Biofortis, Global Specimen Solutions, and Gene Data as well. As you know, those are very strong bioinformatic companies. And he is currently the Chief Technical Officer for a company called Helomics, which is a division of predictive oncology. And using the power of artificial intelligence, which is Mark's backbone and strong suit, Mark is responsible for leveraging Helomics' vast repository of physical and digital tumor samples to build multi-omic predictive models for tumor drug response, which is what he's going to be talking about today. Right? Today, the title of his talk is Driving Drug Efficient, Effective Drug Discovery Using Machine Learning Models of Tumor Drug Response. Right? That's a lot to say about the technical aspect of his talk. On a side note, I believe all of you will be in mute during the seminar, but feel free to type in any questions you might want to ask Mark. There is a Q&A box on the bottom right, which you might be able to access, and you can type in any questions, and the speaker would be able to see those questions as well, besides the panelists. So during the course of the talk, he might be able to answer some of your questions, but the eventual goal is for all of us as panelists to keep a track of these questions and combine them and collate them, so that at the end of the talk, we might be able to address this as a Q&A session. I think that kind of keeps the flow going for the next 40, 45 minutes during the during the talk, and we can jumble everything into one single space as a Q&A session after the talk. And this seminar is being recorded. So a link to this talk would be available soon after. So you might be able to access it, and you might be able to watch it in leisure again as well. So without further ado, I present to you Dr. Collins. Mark, the stage is all yours.
Thank you, Sesha. Right. Hopefully everyone can see my slides. Sesha?
Yeah, I can.
Okay, great. Well, thanks very much for the very nice introduction, Sesha. I do actually remember that interview, and I think, as many of your customers know, certainly Sesha's done a fabulous job of promoting high content too. So I wanted to thank Sesha and the whole Thermo Fisher Helomics team for inviting me to give this talk. I didn't realize I was going to be first, but anyway, here we go. So as Sesha mentioned, I really only had one good idea in my entire career, and that was that somehow, some way, machine learning and artificial intelligence ought to be able to help us discover drugs better. I always wanted to be a doctor, but I didn't get into medical school in the UK for lots of reasons, mostly to do with playing music. And so I spent most of my career trying to help patients get better by discovering drugs or coming up with informatics approaches in order to be able to help do that better. So without further ado, let's get started. So I'm going to give you a little bit of an outline of the talk so you can sort of get the context behind it. So talk a little bit about Helomics here in Pittsburgh. It used to be called Precision Therapeutics. As Sesha said, we're now part of a larger company called Predictive Oncology. Talk a little bit about our clinical offering, which is really the foundation of what I'm going to talk about on the drug discovery side. Talk a little bit about why drugs fail. I'm sure most of you heard all that. Talk a little bit about how we address some of the heterogeneity issues in drug discovery. And we'll talk a little bit about our patient-centric discovery engine. And then I'm going to spend the remainder of the 2/3 of the talk or so, probably more than that, talking about the biology and the high content side of what we do here, and give a little bit of an outlook of where we're going. And then, as Sesha said, some time for questions at the end. So I think everybody has a connection to cancer. And at Helomics, before it was Helomics, it was Precision Therapeutics, as I mentioned, and it really is the core of Helomics is this focus on the patient, right? So cancer's personal. We believe your cancer therapy should be too, and that is what drove the whole process behind Helomics. So the ability to test drugs on patient tumors and use that drug response profile to feed back a recommendation to the patient's clinician in order to individualize that patient's therapy. That means over the last 15 or so years, Helomics has developed a real core competence in testing drugs on primary tumor cultures. And as it's gone along, it's amassed a huge amount of real-world longitudinal data on how those patients of different tumor types responded to drugs. So, when I came on board about 3 years ago, I thought, well, really, if you think about it, what drug discovery is trying to do, what pharma is trying to do is find the right drug for the right patient. And here we have a technology platform and data that has been doing that for the last 15 years. So we leverage that in some way to make drug discovery a lot more patient-centric. So again, as I mentioned, our core is a clinically validated functional tumor profiling test that helps oncologists individualize current oncology therapies for patients. And as I said, that's really a little bit like drug discovery, right? But for the most part, I mean, I've spent a long time in pharma, from like the mid 80s into the early 2000s, and I cured a lot of mice, which wasn't very satisfying because I'd given them cancer in the first place. But in all that time, working in natural products drug discovery and then early days of cell-based assays, we never thought about the patient. The patient was like way down. That was all those clinical folks working with patients, right? So they play almost no part in drug discovery. Yet they're key. I mean, we're trying to treat the patient, And as always, I think one of Sesha's favorite books is the Stephen Covey, 7 Habits of Highly Successful People. And one of those is start with the end in mind. So we really should be starting drug discovery with the end in mind, which is the patient. And we often really don't do that, at least to the level that we need to. So, at Helomics, we now believe that, cancer is personal, but we believe that cancer drug discovery should be too. So we're really trying to bring, using our core competence in patient tumor profiling, drug response profiling, trying to bring that patient much, much earlier in discovery. Well, why do we want to do that? And what really is driving some of the reasons why drugs fail? So drugs fail because heterogeneity, for the most part, right? So that's heterogeneity both within the tumor. We all know the tumor is a very complex ecosystem, if you will, of different cell types, messaging between them and so on. And then heterogeneity between tumors, i.e. between patients. One patient's tumor does not respond the same as another patient's tumor. And, we've spent the last 15 years, essentially doing that, individualizing therapy to the patient. As I mentioned earlier, real patient sample testing just doesn't occur until very late stage in discovery. And if you wanted to exhaustively screen, if you will, compounds against a very large heterogeneous patient population, with existing technologies, it would be prohibitive in time and money, as we'll see in a bit. And again, if you want to take advantage of drug combinations, you really would, again, be really in for a very cost-prohibitive and time-prohibitive approach. The consequence of this heterogeneity, both of the tumor and between the tumor, i.e. the patient, is that over 80% of compounds fail to translate into the clinic. So we firmly believe that if we're able to somehow, as I'll show you in a minute, bring the patient at the heart of discovery, even at the beginning of discovery, then we should have a major impact on improving the chances of success of a compound translating into the clinic. So mostly the ways we deal with heterogeneity, and apologies for my PowerPoint cartoon here, but it's all I could put together in the time. But essentially, we all know how xenografts are built. We take a tumor biopsy. We may well go straight from that tumor biopsy straight into an animal, or we may do an intermediate culture step. But this is really, it deals with heterogeneity, but it's really one animal per compound per dose, which is expensive and time consuming. There are other ways of dealing with heterogeneity, like patient derived organoids, other kinds of models that use zebrafish, transform cell lines with CRISPR, all sorts of things like that. But for the most part, these address heterogeneity in some way, shape, or form, but they're mostly applied at late stage development because of those time and cost constraints, or there's really not enough data around how representative they really are of the patient and tumor heterogeneity. So just flipping across to the sort of financial consequences of doing this, right, this is the standard drug development cost model. Well, we all know it costs a bucket load of money to develop a drug. It cost a lot of money when I worked in pharma, and it costs even more now. But what I want to do is just focus our minds on the hit to lead process, where we think most of the opportunity to bring the patient early would be to apply it in hit to lead, right? It's still a very expensive part of drug discovery, and often late stage failures can be traced back to decisions we either did make or didn't make in hit to lead, right? So being more informed in hit to lead, we think, should be valuable. So if we just look at addressing heterogeneity at hit to lead, so typical hit to lead process, we may be going back, rescreening, our original cell line from the primary screen. Maybe we're doing dose response and maybe some other cell lines or other approaches, but it really doesn't address tumor heterogeneity or patient heterogeneity at all. But it is highly efficient, but it's low information content, even if you did a high content assay. Right? If we take the xenograft model as our gold standard, if you were to do one patient, 100 compounds, cost you about $5 million, would address the tumor heterogeneity, but not patient. It's moderately efficient, again, uses a lot of animals, but you do get more information. In reality, no one would do this. This is the cost prohibitive part of dealing with heterogeneity, right? Is that if we took 100 patients and 100 compounds, it costs us about $500 million to use an awful lot of animals. It would address heterogeneity, but it's low efficiency and high information. As we'll see, our approach, which incorporates, high content, our patient-centric discovery engine can screen hundreds of compounds against thousands of patients highly efficiently and with high information content. So about for the equivalent cost of a single xenograft model, using AI, using a high information readout from high content, using patient tumors, we can really do a really good job, we think, of bringing heterogeneity, bringing the patient much, much earlier in discovery at this hit to lead stage. And what would that mean? Well, obviously it means that we can make better informed decisions earlier in discovery. We can improve efficiency and cost effectiveness, which is great. But most importantly, we can get drugs to patients that need them. And that, for me, is the reason why I joined the pharma industry. And I'm sure many of the reasons why we all do work in this space is to get drugs to patients more quickly and have those drugs be actually useful to them. So I'm going to spend a little bit of time, most of the rest of the time, talking about this patient-centric discovery engine, focusing in on the biology of our tumor model and the way that we generate data from that using high content. I'll spend a little bit of time on the data that we already have, and I won't spend hardly any time on the active learning side. Safe to say that the active learning, which is software we actually licensed from Carnegie Mellon University. Professor Bob Murphy is actually been a long time user of high content, in fact, designed and patented the active learning software. And essentially what it enables us to do is to construct predictive models to drive rounds of experimentation that improve that model. And it really allows us to explore the drug patient space in a very exhaustive way, but without doing too much wet lab experimentation, which obviously makes it quicker and more more cost effective. And really the only tractable way we believe to do this kind of patient-centric drug discovery is to use some kind of active learning approach to actually design the experiments. But I'm going to focus for the rest of the talk on these two parts here, the patient tumor cell line assay, so to give you some insights into that and how we use high content here, and then we'll spend a little bit of time on the public data as well. But the idea behind the discovery engine is really the ability to give back to a customer, for example, results on the diversity of drug responses across a set of patients. Correlations and comparisons of those drug responses with existing standard of care drugs, which of course is the measure for whether you are moving the needle at all for a compound. And to generate models of different drug responses of patients with different tumor properties you can use for a variety of applications, such as companion diagnostic development, biomarker validation, and so on. And then we can also give the results and responses of drug combinations too. So, okay, what's going on here? Okay. So let's take some time to look at the patient tumor cell line assay. So as I mentioned, we have this currently running clinical test. We take data from the patient. We take a functional tumor profile of the patient's tumor. I'll talk about that in a second. We compare those drug response and biomarker profiles, and we feed back a roadmap to the oncologist to individualize therapy. And the platform was clinically validated in ovarian cancer. In other words, we demonstrated that the tumor response profile was predictive and prognostic of the response outcome in humans. But the PDX, the platform itself, the functional tumor profile test, has been used on over 131 different tumor types. So pretty confident that it works for most cancers. Skip over there. Sorry, go back one. Sorry about that. So the patient tumor cell line assay is really, as I've mentioned, clinical utility, is predictive and prognostic, and it's really been running in our lab for the last 15 or so years. It was developed originally by Paul Kornbluth, who was actually a neuro-oncologist, neurosurgeon, and figured that he'd done some work at the NIH on the NCI-60, and then decided to grow tumors and test drugs on them as a way to improve the outcomes for his patients. And from that, this assay was born. There's a whole bunch of patents and papers that you can read on this assay out there in the public domain. But the key things about the assay are the way we prepare the tumor material. That's very vital. And again, there's a bunch of patents that cover that too. But essentially, the assay is a tumor stromal co-culture, for want of a better word, where we maintain cell-to-cell contact as far as possible in the original patient tumor sample. Through a variety of quality control approaches, we can address cellular heterogeneity and make sure that we're representing that original tumor as best as possible. We don't need to make spheroids or organoids, though we can and have done in the past. The most important thing is the test's been running for the last 15 years in a fully regulated, fully automated, and highly standardized manner. And again, we can change the platform for various endpoints. We can measure additional parameters and so on. So from that assay, we use that assay to test thousands of highly well-characterized tumor cell lines derived from de-identified and consented patients. And that is really at the heart of addressing this heterogeneity problem, both within and between tumors. Just to give you a little bit of data on drug response of primary tumor cell cultures. This is 5-fluorouracil, often used in colon cancer, and these are just curves, IC50 curves, essentially of dose versus normalized cell count. You can see the variety of these. Each individual line is a patient. So you see the variety of responses across different tumor types from different patients. This is again cytarabine, again another ovarian cancer drug. Again, see the variety of different responses. Paclitaxel, again another classic Taxol-based drug for use in ovarian cancer. Again, see the variety of response here too. And carboplatin and platinum-based drugs. Platinum-based drugs, as many of you know, were really landmark therapies in the treatment of ovarian cancer. And finally, this is a fairly busy plot. But essentially what we did here was we compared primary tumor cell cultures. Oh, hold on a minute. Sorry. So our offices are super eco-friendly, and the lights just went out because I didn't move around enough. Sorry about that. So this sort of composite plot here just shows how we comparing primary tumor cell cultures with standard ovarian cell lines, HTV77s in this case. In general, the primary cell cultures should say more responsive than the cell lines. Again, representing normal cell count just against either the cell line or a patient sample, and it's against a panel of a subset we're showing here of a panel of known FDA-approved anti-cancer drugs. So really just to summarize the biology of the platform, right? So as we mentioned earlier, classical ways to address heterogeneity or ways that are up and coming, they attempt to replicate the complexity of the tumor microenvironment, but they aren't really at the moment, really useful for research, routine research or clinical use. And if they are, they're very expensive to do, so they get pushed down into late stage usage. We believe that the Helomics True Tumor platform is very cost effective to do. It's high throughput, it's reproducible, it's flexible in measurement capabilities, and it has demonstrated, as I said, clinical utility as representative of the patient. So if you want to bring the patient to the heart of discovery, you better be sure you have an assay that is representative of the patient. Right. So let's dig a little bit into the data, and then I'll spend the rest of the time talking about high content. So we have a lot of real-world longitudinal data on about 150,000 patient tumor cases that span 131 tumor types. And again, all that is both standardized drug response data from that clinical testing, together with biomarker and mutational profiles from a combination of immunohistochemistry, qPCR, and most recently next-generation sequencing. And then because we've been testing so long, and we're a healthcare facility, we have access to over 10 years worth of clinical outcome data. So not only can we, as it were, use our patient platform, we know not only how did their tumor respond to drugs, but we can access, if we don't already know it, how they actually did on the real drug when they were treated, by their oncologist. And again, for about half of these cases, we have access to high-quality archived FFP material that we can do further sequencing on, again, to build up more characterization of the patient tumor cell lines that we're using as part of a drug discovery project. So let's talk a little bit about how we generate models of patient tumor drug response that we then use to drive discovery, to drive the wet lab experimentation. So again, just to reiterate, the engine that we built is a combination of the clinical assay, the representative assay platform, the historical data that informs where to look, and the active learning, which guides the wet lab experimentation in a very efficient, but in a manner that also explores a big drug patient, as it were, space of responses. So we use data and machine learning in lots of ways here at Helomics, both for drug discovery purposes and clinical purposes, but we use data and machine learning to create featurizers of a particular data type. So our models are very multi-omic, so they contain more than just genomics. They contain, in this case, high-content cell data, image data, data from tissue images. We have over 100,000 H&Es, genomic data, so DNA-seq sequencing data, and RNA-seq transcriptome as well, together with the clinical information about the demographics of the patient, age, tumor type, and so on. So I want to focus for the rest of the talk, since this is all about high content. So the data that we have to drive the featurizer, in the case of the cell image featurizer, we have about 4.5 million cell images here from about, like I said, 15 or so years worth of testing, different tumors on some different patients. We have multiple growth metrics, AU7, EC50, IC50, GI50, GI50, and as I mentioned, biomarker and whole exome and whole transcriptome data. So we have a massive database of both physical and, if you will, digital samples. So when I first came on board, I discovered that we have all these cell images. They had a Helomics instrument, which they hadn't actually been using, and we now are using. You'll be glad to know. So we were looking at ways to take the cell images and extract as many features from them as possible to use those features to drive our downstream models of how tumors respond to drugs and how we can then find the best tumor drug response patient relationship, for a particular set of compounds. So we implemented an in-house pipeline only because we need to scale to a huge amount of images and a huge amount of features. So the internal pipeline uses Python. It uses PySpark, which is a cluster method to generate all this data very efficiently. So essentially, we take the cell images. We use a very simple assay, a very simple endpoint assay. In the past, it's simply a DAPI stained image, one field per well from a 384 well plate. Do background correction, do cell segmentation, all classical image technology. Compute cell features, again using very classical techniques. But then what we go ahead and do is compute cell graphs. We felt it was very important, and talking with some of our collaborators and CMU, we felt it was really important to be able to represent spatiality, right? Represent the relationships between cells and how those relationships change in response to drug, particularly because we've got a mixed cell culture in our assay. So we compute a bunch of cell graph features, how close cells are to each other, how connected they are, and so on. And then we compute well features from the cell features, the traditional image analysis features, and then we create a feature vector per image per dose, and then we use that to feed into a bunch of machine learning algorithms to featurize that cell image and use that in downstream models. So along the way, we built a very, very large ovarian cancer graph database from all these individual cell graphs of how cells were related to each other based on different properties. So this is quite an interesting just aside to see the power of graphs in image analysis, and particularly in high content. Cell graphs have been used pretty extensively in tissue, and we in fact use cell graphs in our tissue machine learning projects too. But here we built essentially a small subset, about a third of our ovarian cancer data, 10,000 cases, so 10,000 individual patient tumors, analyzed all the images from those patient tumors, their dose and drug response, which generated, again, 70 million nodes, where we computed a variety of properties, size of the cell, shape, intensity. We have drug treatment and tumor type, and then we have response in terms of all the measures that I mentioned earlier, AUC7 and so on, which generated about 130 million relationships between cells, so that we, as I said, capture spatiality, but more importantly, capture other properties or attempt to learn other properties in the context of spatiality. Right? So just so you know, although it's circular, this does not represent a well. So, you know. So if you just play with visualizing cell graphs, it's kind of interesting. Here's just an example of retrieving about 1,000 nodes, i.e. 1,000 kind of cells, across plates and across wells and across doses. But just looking at responders to carboplatin from those ovarian cancer primary cell cultures, you look at non-responders, you can see some, you know, immediate sort of patterns in that there's much more connectedness between the non-responder and the responder. And again, if you look at the relationships between cell size and spatiality, if you will, we look here at a responder to carboplatin, where we're looking at small cells, and on the right-hand panel here, where we're looking at larger cells responding to carboplatin. So again, we can see much more connectedness in much larger cells than we can against smaller cells. This again is just an aside, just to sort of give you a sense of the richness of the data that we can generate and the value of cell graphs, which we'll come to a little later on. So having extracted all that information from those 4.5 or so million images, focusing just on ovarian cancer for the purposes of this afternoon's talk, we have about 530 features in total, a bunch of what we might call classical image analysis features, including the cell graph features, like the number of components in a cell graph, and so on, how close they are to each other, how related one cell is to another cell, in the context not of the well, but in the context of drug response, dose, and so on. And then we also computed about 450 parallel texture features for the image analysis folks out there. These are classical texture features that look at textures of the cells, essentially. And we evaluated 3 different machine learning models, 3 different machine learning approaches, I should say, to building a model, because what we wanted to do was to take all these features, build a model of drug response to demonstrate that a featurizer, so as it were, a representation of all these, you know, hundreds of features here, was good enough to represent drug response, hence could be used in our downstream models. So we used something called weighted random forest, which is a tree-based approach for vector machines, and standard good old linear regression. We evaluated the best model based on a test data set using root mean square error. That turned out to be the random forest model. So what I'm going to show you now is how we took all those high content features, if you will, and see if we could train a model to predict drug response, and what might be some of the factors driving that model. So first of all, you want to ask the question, well, you know, I computed all these features at vast, you know, complexity and so on. Does it really make any difference? You know, because if it was just turned out to be cell count, it would be kind of it was kind of disappointing. So we asked the question: do all the cell imaging features help in building models? And the answer is so. We looked at computing models using random forest and compared root mean squared error. So the lower the root mean squared error, the better the model's performing at predicting. And we had all cell features, all features except cell count, and only cell count. You can see from the graph, blue is only cell count. So in the majority of cases, in fact, for all the cases, cell count builds models that predict more poorly, less well than either the all features that include cell count or without cell count. It's marginally better actually without cell count than it is with cell count. So we're pretty comfortable then that, you know, all these extra features, going to the trouble of generating them all, And I should say we did this on a small subset before we went and burned a whole load of money at AWS and ran it on everything. But essentially, that's what we did first. So we were pretty confident that our computation of features, made sense in the context of building a better classifier, a better model, I would say. So we then went and explore a little bit, which are some of the features that might be related to dose, doing simple principal components analysis. So we ended up with two, principal components. Variance between those two is listed there. Second principal component, and unfortunately, this is a lot of data, so you can't see the labels so well. But second principal component seems to drive the separation between doses. In other words, these particular features relate to being able to predict, in this case, dose. And again, intensities, which you would expect, cell entropy, how complex the cell texture is, if you will, skew and ketosis, and so on. A bunch of different measures. So then we took all these features with our random forest model, and then ran a whole bunch of experiments looking at to cross-validate whether and how good the model was at predicting, in this case, AUC7, which is our measure of response. So what we had to do before we did that, though, was essentially be sure that we had a good way to represent the patient. So most of our models up to that point were looking at an individual, for want of a better word, in other words, an individual experiment where we had the drug at dose 10 micromolar or something, patient tumor cell, and we had a response based on that. So we developed methods to basically bring together all of the drug responses across the dose range for a particular patient, to then have a model, or to then test that our model could predict for a different, at the patient level, not essentially the well level, if you will. Essentially, the way we approached that, we took a couple of approaches. This one turned out to be the best. So concatenating all the responses, all the features, I should say, for our assay replicates across all doses. And then we use that new feature set to create patient level models on a per drug, per patient basis. As you can see here, that concatenated approach gives us actually really nice results in terms of predicting AUC7 versus actual AUC7. I'll color them here by categorization of responsive versus intermediate versus non-responsive. So again, very happy that the combination of doing exhaustive featurizing of the cell image, taking those features, feeding them into a random forest model, and then concatenating those features together to make a patient specimen level featurizer model that predicts drug response, turns out to be pretty successful. So just to give you a summary of the cell imaging part here. So like I said, I mentioned it's based on multiple features. It takes 431 texture features, 99 of the cell graph features. The model based on a random forest classifier performed best at predicting response. So we're confident that featurization of cell images built good models of patient tumor drug response, and we can then take that featurizer, essentially the representation of all those 500 or so features. It's a machine learning representation, and we can use that for our overall model building approach, together with all the other featurized data types, transcriptomic, genomic, and tissue-omic data spaces as well. So one of the things that we're doing in future directions, and I just want to give a little bit of a shout out to both Brent and Nick Radio, and particularly on Kenwick, who managed to get a very old Generation 1 Cell Insight to work. As I mentioned, maybe I didn't mention it, but when I arrived first at Helomics, they had built their own images, which was kind of startling, considering that we were literally across the river from Helomics when I worked at Helomics, Thermo Fisher. So we've now actually been validating that cell insight for use in our clinical assay versus the home-built images that had been built 10, 15 years ago. And we're starting to investigate richer phenotypes, so we're looking at cell painting as a way to generate a richer phenotype, to generate an even richer feature set that we think will help building better models of patient tumor drug response based on the high content imaging approach. And we're also expanding the drug panel that we test on. I mean, while we wouldn't do it because it's kind of expensive, you could imagine, essentially building yourself which interestingly was actually one of Helomics' original ideas, was to build a gigantic database of phenotypes in response to different drugs, using, in this case, patient tumor cell lines, not immortalized cell lines. You could build yourself up a very comprehensive data set, which again you could use for further downstream usage. I spent a couple of minutes wrapping it all up together, getting close to the 2 o'clock hour. So when we think about running this patient-centric discovery engine, right? So as I mentioned, we have this patient, a tumor assay for testing drugs on patient on thousands of heterogeneous, well-characterized tumor cell lines. And we have the data to inform us where to look. And we have, as I mentioned, the active learning to be able to exhaustively explore in a very tractable, very efficient manner a big patient drug response space by guided selection of experiments. So let me show you kind of how that works. So the patient discovery engine process brings all those three elements, that sort of trifecta of the assay, the historical data, and the active learning together to actually drive a hit to lead campaign, right? Where we're bringing this patient into the heart of that hit to lead campaign by using that patient assay and those patient cell lines. So essentially, the way the engine turns is it does iterative cycles of learning, predicting, and testing. So we essentially build initially a set of models, and really what the active learning, the core platform does, is it builds models and it tests how uncertain it is about that model as to how good it'll make the overall model, and then attempts to, but then gives essentially a list of experiments that would be the most informative to improving the overall model. Most models work with essentially static data, right? They're just trying to improve predictions based on a static data set. What we're doing in the discovery engine is doing real experiments guided by the machine learning, guided by the active learning, to generate data that will be most informative for making a model that optimizes the relation between a drug and a particular kind of patient, depending on whatever our stopping rule is here. So we constantly cycle round, doing some learning, doing some data generation, testing compounds on particular subsets of patient tumor cell lines, updating the model and iterating around until, as we said, we reach our stopping criteria, which might be we have 3 or 4 lead candidates to which these particular different patients, represented by their cell lines, respond to optimally. And then we can go ahead and do further characterization on those patient tumors if we want, and further data generation to more deeply characterize them, if we will, or we can carry on and do additional experiments. So just to summarize the overall thing, we've been testing tumors using high content and cell imaging for the last 15 or so years. And we have a clinical test based on that cell imaging that tests the drug response of patient tumors and guides clinicians in individualizing patient therapy. We've now taken that core competence and the data together with some proven AI in the form of this active learning technology to really create what we believe is a unique capability to do oncology drug discovery, where we can highly efficiently screen drug responses against thousands of diverse, well-characterized cancer primary tumor cell cultures. And as I've convinced you of, it's ideally suited to the hit to lead stage and should result in better prioritization of compounds and better coverage of patient diversity earlier in the discovery process. And obviously, we strongly believe that that's got to be better than not involving the patient, and that should definitely improve the chance of translating discoveries into the clinic. And as I mentioned, we make a lot of use of high content, a lot of use of machine learning on cell images, and we intend to make even more use of that. Now we have our Cell Insight all up and running, and who knows what else we'll do in the future. So I've got a bunch of people to thank. My team here at Helomics on the bioinformatics and machine learning and data science side. I had a couple of CMU interns this year who did some very nice work on some of the tissue and genomic stuff. Collaborators at Carnegie Mellon, Professor Robert Murphy and Dr. Josh Kangas, who both advise on the general machine learning side, but also the active learning side. Some of the work that I've described here is in collaboration with UPMC McGee-Women's Hospital, where we have a currently running study using this machine learning approach actually to build on the clinical side an improved AI-driven clinical decision support system that will predict which drug you should put the patient on and what the prognosis of the patient on that drug will be. So that project is slated to finish at the end of this year. And a lot of advice always from a big fan of high content, Dr. Paul Johnson at the University of Pittsburgh. Did a little bit of organoid and spheroid work with his lab, and that was really quite interesting. And then from the thermo side, as I mentioned, shout outs to Brent and Nick, and particularly Leon, who came to our site in the middle of lockdown and helped diagnose what was wrong with the Cell Insight. So thank you again all very much. I'm happy to take questions.
That was an awesome talk, Mark. Really appreciate it. I wonder if there are any questions from the participants. Please feel free to type in. You can, Laurie, I'm not sure whether they can unmute themselves and ask questions.
No, they need to just type in the chat.
Okay. I have a few questions of my own, Mark, if you don't mind.
Sure.
I think AI and machine learning have always been of interest personally to me at some level. It seems like that's where the technology is going as far as data crunching goes, especially with the stuff that Broad Institute has been doing. And commercializing a solution like this is definitely something that is going to be the highlight in the next several years. But that said, it seems like ever since Peter O'Brien and Jim Zhu had published a few papers on seeing the value of the multiparametric nature of the high content assay with their papers, right? Back in back in 2006 and 2008, it seems like nothing has come through in the forefront that is close to what you are trying to accomplish, right? I mean, their classic papers clearly suggested the value of extrapolating a cell-based assay to a clinical situation, right? And you've gone one step further by using Python and machine learning algorithms to kind of crunch the numbers at a different level, right? I think you might have answered the question in the last slide based on the work that you're doing with Paul Johnston's right now. My thought was to see if there's any benefit into looking at these things, not in just one dimension, but getting into the cell painting space, getting into multi-dimensional structures like organoids, for example, especially organoids from patients who have been treated with cisplatin, carboplatin, and all those drugs. You see the benefit of that? That was one of my questions. And how are you going to use machine learning algorithms to kind of build on top of what you already have? It's going to get complicated, right? Data crunching. I wanted to see what your thoughts are on that.
Yeah, I mean, I think, I mean, just to go back to the sort of comment you made about the Peter O'Brien and the DILI stuff and multi-parameter cytotox and all those other sorts of things, it was certainly, shown to be predictive, in man. However, what you were using there, were, essentially, were either primary cells, liver primary cells. Again, I think what it comes down to is certainly, I sort of give you some background from my career. I mean, I've either had all of the data and none of the tools. I've either had the machine learning and none of the data and none of the biology either, or I've had the biology, but none of the data and none of the tools. So I think what it comes down to is what what O'Brien and lots of other people have demonstrated. There's incredible value in measuring a lot of things, right? But you got to measure a lot of things on the right thing, right? So moduli cell line, yeah, go ahead. I think you have to have that union of the right model, and it's not really a model either. In our case, it's not a model, it's the patient cell, tumor cells, right? It's not a model of the patient tumor cells, it's the patient tumor cells. Plus all that historical data that you can use to inform you where to not look, right? Sometimes, people say, I'm an expert is someone who's made all the mistakes. So I think it's that union of three things. Having a representative approach to testing patients, a surrogate, if you will, for the patients, as we go and actually test on patients. The historical data that drives, you to go look in the right place, and the appropriate use of machine learning to get you where you need to go to. As to whether spheroids, organoids, without a doubt, I'm not saying they're not a good idea. They certainly are, and there's certainly you can point to very interesting patterns of drug response compared to when you compare cell lines. When you compare spheroids and organoids generating cell lines from patient primary cells and so on. It's just that from our perspective, right, since we're making a whole point about how the in vitro model is predictive in man, we're not quite there yet with spheroids and organoids. We'll get there, I'm pretty sure. there's several companies out there trying to build up banks of patient-derived organoids for use in screening, but it's at the moment we're taking the approach that those three things together, are really what's needed. Yes, we could use spheroids and organoids, but we don't have enough data on those yet ourselves, and we don't have enough data generally on their use, in that clinical setting. So I think it will get there, and machine learning will always play a part, right? Because we're going to end up with a lot of data to sift through and to essentially compress down into some sort of compressed feature set. I mean, if we went ahead and used every feature that we extracted from our cell images, it wouldn't help you. So we have to compress down, which includes usually machine learning techniques to do that.
Yeah, I mean, that's a great thought to end with as well. I think even if we can show drugs that have been taken off the market for some reason, and see that they have a certain signature versus drugs that have been implemented in the market by FDA approvals and standard processes, and show the key differentials, right? The servastatin versus pravastatin study that was done many years ago, something like that, right?
Yeah,
that is kind of going to tighten your data.
Yeah, I mean, I think a very wise guy said to me, a very wise person said to me once, we probably have all the drugs we need. We just don't know which patient they work on best. And that's essentially, is where I would go and look for. Go and look at all the compounds that didn't make it through the clinic because the patient wasn't involved till late in discovery, and repurpose those that hit the lead using a patient-centric approach. It's an obvious thing to do.
Absolutely, no question about it. But I mean, this is like the forefront of where technology is actually going in terms of high content. I see a true value in what you're accomplishing right now. I think this is awesome. I mean, do you see this extrapolatable to other disease states like COVID, for example, right?
Well, the active learning part for sure. Yeah. So if you wanted to do the active learning doesn't really care what it's trying to what experiments it's trying to use, and that's actually been used in things like on the public ToxCast data, where it was shown to be able to learn in the hits from the ToxCast data set in a much more efficient manner, so less rounds of experimentation, wet lab experimentation. So certainly, several companies out there implementing active learning approaches of different types in order to be able to discover treatments against all kinds of diseases. There's companies out there. There's actually something called the ATOM Consortium, which is a GSK, NIH, Lawrence Livermore sort of consortium of people using active learning. But theirs is more chemocentric, right? So they're looking for, using active learning to explore a massive space, a massive chemical space, to see if that will hit a particular target. And I think a company called In Silico Medicine down in Baltimore that found some COVID-19, some drugs against the spike protein that would using active learning and machine learning approaches in trying to, again, very target centric or very chemocentric. What we're trying to do is be very patient centric, because that's where we believe you can have the most impact, and particularly if you can have that most impact early in discovery. The earlier in discovery, the better.
Yeah, 100% agreed with that. I think this was an awesome talk. Go ahead.
Mark, this is Matt.
Hey,
Matt.
We
have a couple questions in the chat here that I wanted to get addressed before everybody jumps off. There's A question, are featurizers equivalent to a set of descriptors from your machine learning model? And then the second question, what's the requirement of imaging acquisition if we want to use them for machine learning? So 2 questions.
Right. So yes, to be very technical, what the featurizer is essentially latent featuring as the latent layer of a machine learning model. So it's essentially compress those features down, and you're giving the downstream, machine learning a model, for want of a better word, of all the features that you input to the featurizer. So that's, it's a subset of the features which best describe, in our case, drug response. But it's actually a layer from the machine learning platform itself. And then in terms of image acquisition, I mean, I think there was always this debate when I worked at Helomics, right? Was how good does image have to be? How do you want 10x? What's the magnification? How good do you need the optics to be, et cetera, et cetera. I think that imaging systems these days are so good that pretty much any imager, any fluorescent imager is going to give you more than a good enough image in order to extract features from. But again, you've got to be careful of things like background correction. You've got to be careful of things about how your image is in focus. I mean, a whole bunch of things. We implemented pipelines that did a whole lot of quality checks on those images. Again, when you build machine learning models out of data, you've got to be really, really cognizant of all the things that can impact that. So your background correction, the way you're segmenting cells, methods used for segmenting cells. Again, these are known problems, right? You just have to take them into account. It's not like we don't have good ways to segment cells anymore, or that we need to develop. So I think it's look at the quality of your images, look at the various best practices on how you generate features from images, and then again downstream of that, whole best practices in how you construct and design and test and validate machine learning algorithms too. It's so easy to do it now that you can get into some bad habits. We see a lot of we read a lot of papers and we see a lot of overuse of data for staff. So.
Dr. Collins, this is Victoria. Thank you so much for your talk. I also had a question. It's more on a clinical side. I'm very inspired by the work you're doing. Just kind of like you mentioned earlier, every single one of us has somebody, that had cancer, that's battling cancer, and this has, affected my family. So one of the things I was just interested in, when a person goes through this kind of tragic treatment and event, it's very hard on the family and everyone. But what I've noticed through the experience we had in our family is that it was very difficult to find this kind of service for patients that have, rare cancers, or the cancer is reoccurring, and, the physician might not know what the best, steps moving forward. So to me, it looked like the access to this kind of service is very limited, right? So how, in your experience, what are you seeing from the clinicians? Are they open and they're embracing, this individualized approach to patient therapy? And, what do you see the biggest challenge getting the physicians actually to take those reports and to, to use that data as part of the treatment plan, if that makes sense?
Yeah. So, I mean, I think in the assay that we have for ovarian cancer has been running very successfully here for a number of years, and we've helped. We have several hundred oncologists who who have sent tumors in the past. I think there was a little bit of a how do I put it? It seems a dated technology, right? Testing tumors, testing drugs on tumors, because everyone sort of, I guess, in the late mid 2000s is like, well, we can just deal with this with mid 2010s, I would say. We can just do this with genomics. Look, we'll just sequence the patient tumor, and we're off to the races, sort of thing. But the reality is that while I can sequence your patient's tumor, and many patients' tumors do get sent for sequencing at Foundation or whoever it might be, for the most part, I can't do much because most of the variants that are identified in the tumor don't have an actionable outcome, so we can't tie a drug to it, right? We all know the stats on how few patients get matched with targeted therapies based on genomic profiles, and even then how few of those patients actually respond when they do get the targeted therapy. So it's clearly something beyond genomics, and I think what we're starting to see is a couple of companies. I was at an AACR meeting last year, a couple of companies starting to sort of say, hey, we should do functional tumor profiling. And so I put up my hand and I say, yeah, we've been doing that. The company I work for has been doing it the last 15 years. So I think it's oncologists are always looking for actionable information. I think that's what we can provide them with. And again, I don't, I can't say anything about the clinical side, but visit our website and all the information for patients, and what's needed and how to ask your oncologist and so on, is there. But I think we're certainly talking to our McGee collaborators, and part of the reason why we're building this AI-driven clinical decision support system in collaboration with them is to improve the way that we can take lots of different data sources, lots of different aspects of profiling the tumor, including the functional tumor profile of the drug response, and be able to use that to give a much, much better prognosis for patients. Because most clinicians realize that do a genomic test, you get a very weighty, long report back, but for the most part, for most patients, it does not help them, right? It doesn't say, oh yeah, give you this drug. And particularly in Ovarian, where we have no really good biomarkers at all, still, it is for ovarian cancer, functional tumor profiling is without a doubt one of the better ways forward, because we just don't have good genomic or other inclusion criteria, for recommending a therapy.
Thank you so much for that.
You're very welcome.